There are 4 different forms of albinism: OCA1, OCA2, OCA3, and OCA4. These are caused by the TYR, OCA2, TRYP1, and SLC45A2 genes respectively. The most severe form of albinism is OCA1. Note that OCA1 can further be broken down into OCA1A and OCA1B. The differences between these two is that those with OCA1A have a complete lack of pigmentation, while those with OCA1B have minimal pigmentation. The overall purpose of this study was to estimate the relative frequencies of OCA1 and OCA4 based on the ethnicity in Korea, as these are the 2 most prevalent forms of albinism in this geographical area. In order to do this, they enrolled 12 patients whose age ranged from 0.8 to 29.9 years old, and their parents into the study. 5 of these patients had a complete lack of pigmentation, while 7 of these patients had a minimal to moderate pigmentation. The main technique that was used in this study was polymerase chain reaction, also known as PCR. On this slide, I have listed the components and the volumes of these components that were necessary for this reaction to occur. I will go through each material individually and explain their role in this reaction. First, the genomic DNA template is simply a purified version of the DNA sample obtained from the patient. It is a TYR sequences of this template that will be amplified in the reaction. Second, the primers are made up of specific sequences of nucleotides that bind complementary to the DNA template. These primers actually come in pairs that consist of forward primer and a reverse primer. These primers bind to opposite ends of the template DNA so that they can build the target piece of DNA in the 5' to 3' direction. Third, the DNTPs are simply the building blocks of new strands of DNA. Fourth, the PCR buffer maintains the chemical environment needed for the reaction to proceed. And lastly, the Taq DNA polymerase is the enzyme that actually makes the new strand of DNA. As you will see in the next slide, it is important that Taq polymerase is used in this reaction because it can withstand the drastic changes in temperature that occur. Now that we know the materials that are needed for this reaction, I am now going to explain how the process of PCR works. The first step is denaturation. As in DNA replication, the 2 strands of DNA in the DNA double helix need to be separated. The separation happens by raising the temperature of the mixture, causing the hydrogen bonds between complementary DNA strands to break. The second step is annealing. Primers bind to the target DNA sequences and initiate polymerization. This can occur once the temperature of the solution has been lowered. Note that 1 primer binds to each strand. The third step is extension. New strands of DNA are made by the template DNA. The Taq polymerase enzyme joins the DNTPs together. The order in which these DNTPs are added is determined by the sequence of nucleotides in the template DNA strand. The result of one cycle of PCR is 2 double-stranded sequences of target DNA, each containing one newly made strand and 1 original strand. The last step is the final extension. This allows for the re-annealing of the PCR product into double-stranded DNA so that it can be visualized using ethidium bromide after gel electrophoresis. The ethidium bromide allows scientists to see if a mutation was present by comparing the bands created by the PCR product to a control group. The control group in this study was the patient's healthy parents. PCR was used to amplify the entire TYR gene, which included all 5 coding exons and intronic flanking regions. This is the reason that 6 different primer pairs were required for this reaction. Primers are made to bind onto a specific sequence of DNA, or in this case, to a specific sequence that will amplify a mutation. The use of 6 specific primer pairs allowed the scientists to locate what exon the TYR mutation or mutations occurred. If only one primer pair was used, it would be almost impossible to locate a mutated sequence. This is because there are sequences within the gene that are very similar or even identical, so it is possible that a primer could bind to a sequence of DNA that would not lead to the amplification of a mutation. This could cause some confusion as to whether or not a mutation is actually present. The same goes for the SLC45A2 gene. PCR was used to amplify the entire gene, which included all 7 coding exons and intronic flanking regions. 10 specific primer pairs were used to amplify specific mutation sequences so that the mutations could be identified within the gene. The data here provides the patient's age, gender, diagnosis, causing gene, mutation, and clinical phenotype. I will quickly run through each of these categories for patient 1 for further understanding. At the top, we have patient 1, who is a 2.2-year-old male. He has been diagnosed with OCA1A, which is caused by the mutation of the TYR gene. We can see here under the mutation column that both of his parents have an insertion of an extra cytosine at the 929th base pair of their coding DNA. This is important to note because this is an autosomal recessive disease, which means each parent must have at least one gene that has a mutation that leads to albinism. This individual also has white hair and skin, as well as nystagmus, which is simply a vision condition that causes uncontrollable eye movement. Moving down the list, we see that overall patients 1 through 8 are essentially the same. However, we know that OCA1B is associated with minimal amounts of melanin, not total. So it makes sense that the hair color of those with OCA1B is blonde instead of white. As for the mutations, in this study, mutations in the TYR and the SLC45A2 genes were looked for, while mutations in the OCA2 and the TYRP genes were not. This is most likely the reason that some of the mutations were unable to be identified. The data shown here provides the TYR mutation's location, nucleotide change, amino acid change, and number of alleles identified. Notice that there are 6 distinct mutations consisting of 4 missense, 1 frameshift, and 1 splice site, and 15 alleles of 8 OCA1 patients. Again, I will run through each of these categories for the first TYR mutation for further understanding. In the first row, we have a TYR mutation that has taken place on exon 1. It is caused by the replacement of guanine with thymine at the 155th base pair of their coding DNA. According to the last 3 columns, this mutation was only found once. This mutation was found in only 1 allele of 1 patient. Overall, it was determined by the results of this study that out of the 6 alleles, C929INSC was the most frequently detected in Korean patients and was mainly associated with OCA1A phenotypes.